I am going to kind of highlight and write some stuff, but mainly I'm going to like cross it out so we covered it. So first, this chunk, know your primary and secondary digestive organs, know which organs comprise alimentary tract. You can do it, it's not a big deal, okay? So I'm not going to go over the parts. Got it? Be able to tell this. So membrane is serous membrane. And know that visceral peritoneum covers the organs, parietal peritoneum covers the walls of the cavity. Okay? I'm not going to ask you which parts of alimentary tract are retroperitoneal or peritoneal. You're not going to get the question something like transverse colon is what? Retroperitoneal, peritoneal? No, don't worry about it. You do need to know that organs that are located posterior peritoneal cavity are retroperitoneal. Mesentery, blood supply, immune responses. Main digestive activities. I expand this right here. What kind of question you can get about the digestive activities? If you want, I can bring the bring the lights down. So it's, I know that it's sometimes hard to see. No, but if you promise not to fall asleep. So, digestive activities. One type of question can be, what is mastication? Chewing. Okay. Another type of question can be, which digestive activity from the following list is carried out in the mouth, in the stomach, in the intestine? That makes sense? Nothing trippy, but like if you try to convince me that you carry out mastication in your stomach, some pretty remarkable anatomical. Okay? So just, reasonable stuff. So like propulsion. Again, peristalsis and deglutition, right? Chemical digestion. Salivation, absorption, that's your chemical digestion. Mechanical breakdown, mastication, segmentation, churning, and that kind of stuff. Usually these are the trippy questions. So external stimuli, just know what external or internal. If I ask you, stomach distension, that's internal. Smell of food, that's external. Am I clear? Does that make sense? Like change in the pH, internal. Also remember, you do not really appreciate consciously your internal stimuli, like change osmolality or change in pH in the stomach. You normally don't know about this. Does that make sense? External, you do. Which type of reflexes? Come on. Autonomic, speak up, please, speak up. Autonomic reflexes. All reflexes of digestive system are autonomic. And effectors are smooth muscles and glands. Endpoint effects. Food breakdown. Okay. Internal stimuli. We talked about various from distension to change in pH, change in osmolality, presence of certain chemicals. Essentially, you can have chemical stimuli and mechanical stimuli. So we can have chemoreceptors and mechanoreceptors. Okay? That's what we're talking about. Chemical and mechanical. So sensory inputs will either reach CNS. Remember, we had this flow chart of digestive reflexes. So reflexes can be mediated by central nervous system, spinal cord, brainstem, or they can be mediated by local gut brain, which is located usually in the submucosa of the alimentary tract wall. 4 layers. picture. Okay. I'm gonna slow down. Slow me down if not keeping it up. It's all recorded, by the way. Yes.
You said what about the gut brain?
It's in the submucosa of alimentary tract wall. Look, you have to know that gut-brain, it isn't just floating in your abdominal cavity. That's actually in the wall of alimentary tract. It's not some chunk of neurons that's sitting next to the stomach, for instance. Okay. 4 layers commonly found in the walls of alimentary tract. That mucosa muscularis externa and serosa. All right. What you need to know about those layers. I call it memorization question. What's the function of muscularis externa? Peristalsis and churning, segmentation. Where can be found lamina propria? Mucosa. Okay. What secretion does serosa produce? Serous secretions. You'll be surprised. When I say what type of tissue, like connective tissue, a bunch of different ones from muscular to connective to epithelial and mucosa. Muscle tissue and muscularis externa, usually 2 layers. I'm gonna give you a general idea. So, main digestive activities that occur in the mouth, that harkens back to what I told you about digestive activities. Know what happens where? Hard and soft, uvula. Uvula closes your nose, tonsils encounter pathogens. Know the difference between intrinsic and extrinsic salivary glands. Intrinsic is single-celled, extrinsic is big. Remember, intrinsic salivary glands release lingual lipase. Okay. Extrinsic produce amylase. Now, submandibular most saliva, rotted is the biggest. So this is something like, functions of the components. Mucin, sticky part. Water, dissolves food chemicals. Lysozyme and immunoglobulins, protection against pathogens. Lingual lipase, breakdown of fat. Salivary amylase, breakdown of carbohydrates. 2 types of secretions. The saliva is made of serous secretion, watery, and mucus. Remember, so these are autonomic reflexes. So when I say which branch of the nervous system, I'm talking about sympathetic, parasympathetic. Parasympathetic stimulates, sympathetic inhibits. Salivation reflex. Food in the mouth, sensory input to the medulla oblongata, stimulation of salivary center, motor output to salivary glands, which release saliva. So far. Okay, when you're excited, sympathetic system kicks in, constriction of the blood vessels, less saliva produced, thick saliva. Salivary glands are removed. What happens to your ability to digest or swallow food? It's reduced. No mucin produced, Teeth will start to rot if salivary glands have been surgically removed, because saliva cleans teeth. What peritonitis is? It's an infection of salivary glands which is caused by the virus, complication for males, testicular inflammation. Prevention is vaccines. For the tooth, mainly, so for tooth, I'm gonna do like this part. 3 parts: crown, neck, root. layers: enamel, dentin, pulp. Know that blood vessels and nerves are in the pulp cavity. All right. Okay. Makes sense so far? Gums, connective tissue covered with stratified squamous epithelium. 20 deciduous, 32 permanent. Difference: permanent to permanent, deciduous to temporary. Molars, premolars, I'm sorry, are not present among deciduous. Now for pharynx, I'm sorry. 3 parts, know them: naso, oro, laryngopharynx. Know which type of epithelium covers each. Nasopharynx is pseudostratified columnar, oro and laryngostratified squamous. Also, if I tell you from the mouth food enters what? Tell me. Oropharynx, okay? Not nasopharynx, not laryngopharynx, oropharynx first. Now, swallowing reflex. Remember those couple pictures that we had with the swallowing? That one full slide picture with a fella swallowing food. Know the steps. So you're pushing the bolus down the throat. Signal goes into the medulla, generates the motor response. Uvula closes nasopharynx. Epiglottis closes larynx. Upper esophageal sphincter opens, and food is moved into the esophagus. Does that make sense? All those steps. So esophagus joins stomach at cardia. They are separated by lower esophageal or gastroesophageal sphincter. That's that. Esophagus still stratified squamous. Protects against rubbing. Muscularis Externa, skeletal muscle on top, smooth at the bottom of esophagus, kind of change in between. Esophagus doesn't have serosa. It's right here. Okay. Now remember, we talked about esophagus-stomach junction, how abruptly the epithelium changes from stratified squamous to simple columnar. Am I clear? So, distribution of muscles over the esophagus, skeletal, smooth, mix in between, and stratified squamous epithelium, okay? Anatomical parts of the stomach. You can do it. I'm talking about fundus, cardia, body, pyloric antrum, pyloric canal, right? Functions. Now, this is something that we've done in the quiz, and I want you to know that if I ask you, like, where are the main digestive activities carried out, you tell me that's fundus and body. Okay, so where acid is produced? Fundus and body. Where is pepsin produced? Fundus and body. Cool? Okay. Muscularis Externa, remember, 3 layers. This one, which other layers can you find in the walls of the stomach? That's the same as alimentary tract. Now, epithelium. Simple columnar. In terms of types of cells, what is the function of parietal cells? What do they produce? Hydrochloric acid. What's the function of ECL cells? Produce histamine. Those are going to be kinds of questions that you can see. Does that make sense? We're good? So that kind of highlighting, this question's highlighting what I'm asking here. A vitamin that parietal cells absorb is vitamin B12. So epithelial composition between body and fundus, cardia and pylorus. So remember that gastrin secretion is mainly in the pylorus, like pyloric antrum, pyloric canal, while hydrochloric acid and pepsin secretion are in fundus and in the body. Does that make sense? Okay. Now, regulation of, that's what we just talked about. Gastrin secretion, know the steps. So, gastrin stimulates histamine. Histamine stimulates hydrochloric acid release. So, gastrin from G cells stimulates release of histamine from ECL cells, which stimulates the release of hydrochloric acid from parietal cells. Know this very well. There are going to be questions about it. Effects of gastrin secretion on the stomach, small intestine, ileocecal valve, large intestine. So in the stomach, it stimulates secretory activities and churning. In the small intestine, it stimulates peristalsis. It opens the ileocecal valve and stimulates mass movements in the large intestine. Okay? So ghrelin, you hungry? No food in the stomach. Ghrelin is released, you feel hungry. Serotonin. Food stimulates release of serotonin. An effect on the stomach. Remember, serotonin increases the muscle activity. Okay, smooth muscle activity in the stomach. Histamine, released by ECL cells that are stimulated by gastrin. So histamine activates parietal cells, which produce hydrochloric acid. Histamine, antihistamine OD, like Benadryl, inhibits digestive processes in the stomach. Somatostatin has inhibitory effects on the stomach activities. That's basically as far as it goes. So if I ask you, what effect does somatostatin have on stomach secretion? You tell me. Reduces it. On the stomach motility, reduces it. Somatostatin in this case is inhibitory hormone. Does that make sense? Serotonin and histamine and gastrin indirectly are stimulatory hormones. Okay, so this one. Remember that picture of the parietal cell with carbonic acid dissociating into bicarbon hydrogen ion? That makes sense? I will ask you about this process. I will not ask you about how bicarbonate gets into the blood. But I can ask you about, like, hydrogen. How does it get in the lumen? Active transport. Where does it come from, this hydrogen? From dissociation of carbonic acid. Does that make sense to you? Mechanical digestion, just know that it's churning the chyme and not all the stomach content goes into the duodenum. Transport of chyme into the duodenum, pyloric sphincter. When chyme gets into the duodenum, now remember there are two types of effects, the short term and the long term. In the short term perspective, chyme in the duodenum increases gastric activities. Okay? Does that make sense? In short-term perspective, chyme in the duodenum increases gastric activities. In the long-term perspective, it decreases them. Now, I'm going to generalize here to some extent. Remember, I showed you a giant scheme with gastric, cephalic, and intestinal phase, and stimuli that can be inhibitory and stimuli that can be excitatory. Remember those? Remember that scheme? No? No? It has a stomach in the center, and you filled out the table in one of the labs. So what I'm referring to, you have a stomach in the center. Okay, that's kind of a poorly defined stomach. Okay. And then you have cephalic phase, gastric, and intestinal. And they're stimulate each phase. Like, give me an example of cephalic phase stimulus. Huh? Senses, yeah, smell, taste of food. Give me an example of gastric phase stimulus. Distension, good. Intestinal. Well, gastrin that comes from the intestine in response to what being in intestine? Chem, yeah. Chem in the intestine, that's your intestinal stimulus. Does that make sense? So, I want you to understand which of the stimuli that I'm going to list on the question belongs to where. So, you know, if I smell, it's not stomach. Something like that. And whether the stimulus is inhibitory or excitatory. So, increases digestive activities in the stomach or decreases. So, if I ask, you know, smell of food, you tell me, oh, that's cephalic phase, and it increases. It's excitatory stimulus, okay? If I tell you increased acid in the intestine, tell me that's gastric, and that's inhibitory. That makes sense? Am I clear? Now, sympathetic stimulation inhibits gastric activities. So basically, for some reason, I asked this question twice. Yeah, I asked this question twice. I probably didn't delete this one before. So, in the intestines, as we mentioned, in the intestines, you need to know, like, you know, stretch of the intestine in the long-term perspective will inhibit gastric activities, right? That refers to chem in the duodenum. So in the long-term perspective, stretch of the intestines and presence of food in the intestines will inhibit gastric activities. In the short-term perspective, it will stimulate it, but in the long-term, inhibit.
Isn't that kind of like the exact same question up top?
The what? Yeah. Yeah, thank you. Yeah. It just more, I broke it down into several. So like this top question just tells you, oh, what effect does chem in duodenum? But this one is broken down in the sense of chemical changes, presence of food, mechanical changes, stretch. Does that make sense? Same goes, inhibition of digestive activities in the intestinal phase, sympathetic stimulation. Gastric ulcer, Helicobacter pylori. Know that gastric ulcer is caused mainly by Helicobacter pylori. Okay. And gastritis. Duodenum. Well, know which organs empty in the duodenum. Nerves controlling duodenal activities are autonomic. Three structures I refer to: circular folds, villi, and microvilli. That's a columnar epithelium. Microvilli are folds of the membrane. Laminopropria, as we mentioned in the mucosal layer. Mold protects against pathogens. Lacteal absorbs fats. That is brush border, which harbors enzymes that I just removed from the whiteboard. Intestinal glands secrete mucus and what we call juice, cells that can be found there. I kind of list them: goblet cells, enteroendocrine cells, Paneth cells, and they're mucus. So, and they're absorptive cells. I'm sorry. Okay, so know their function. Brunner's gland produces alkaline mucus that neutralizes acid that comes from the stomach into the duodenum. Now, I'm going to skip this because we did this just today. And I will ask you questions about, what these enzymes do. Okay? Now, in terms of secretin and CCK, know that they're produced by intestines. Secretin in response, and regulation. Secretin is produced in response to acid. CCK in response to fats. Secretin stimulates the release of bicarbonate from pancreas and bile from liver. CCK stimulates the release of enzymes from pancreas and contraction of gallbladder. All right? So we have this scheme where we show how they regulate digestive activities. Know that scheme. Okay, there will be questions about it.
Contraction of gallbladder and what was the other thing?
You're talking about CCK? Yeah. Release of enzymes from pancreas. So that's what effect does CCK have on pancreatic secretions? Well, this is more of a think through idea. So CCK stimulated by fats, so it immediately follows up the stimulation with a contraction of gallbladder, so bile salts in the bile can start emulsifying fats. Does that make sense? So always think about it. Hormone elicits the effects that counteract the stimulus. Mechanical digestion, segmentation. Liver. This is an anatomy. 4 lobes, attached to diaphragm, anterior wall of abdominal cavity. This part. Liver lobes, hepatocytes, and Kupffer cells, macrophages. Know the function. Portal arteries and veins bring blood to the liver, and blood's mixed in the sinusoids. Sinusoids enable exchange with liver tissue because they're really permeable. Know the path of blood. So basically, of portal veins and arteries, sinusoids, central vein, hepatic vein, vena cava. We mapped it out on the slide right there. It's on the video. Okay? But we mapped it out during the class. So it's vena cava. Cell produce bile. Hepatocytes. Bile is released in the bile ductals, collected by common hepatic duct, which then merges with cystic duct, forming the common bile duct. So know the functions of these chemicals. Like, where does bilirubin come? Bilirubin comes from breakdown of red blood cells. Where does bile salt come? Where do bile salts come from? They come from liver to emulsify lipids. What's the role for cholesterol? It's a product of excretion of steroid hormones. Okay. Know the features of bile salts. They're amphiphilic, meaning that they are both hydrophobic and hydrophilic at different ends. So this is why they can emulsify lipids. And these assemblies of bile salts and lipids are micelles. Kidneys cannot excrete bilirubin because it is bound to albumin. So bilirubin in the intestines is processed by bacteria, forming urobilinogen. And urobilinogen then may either stay in feces or enter bloodstream through enterohepatic circulation. And then, so basically, if I ask you urobilinogen, can it be excreted by kidneys? Yes, it can be. Okay. Detoxification, it does it. That's the level that I want you to know. It may chemically convert dangerous chemicals into something more benign. Kupffer cells are macrophages that destroy pathogens. Carbohydrate metabolism, conversion, everything to glucose, conversion of glucose to glycogen. Okay. Lipid metabolism. Lipogenesis happens in the liver very actively. Synthesis of lipids if glucose is in excess. Major proteins. Basically, you can answer to this question with blood proteins. Namely, liver is critical for synthesis of albumin, and which is the main protein of the blood, and coagulation factors, coagulation proteins. Okay, got it? Intrahepatic circulation, we just answered this question today between liver and intestines. So bile salts, they get reabsorbed from the intestines, go into the liver, stimulate more bile salts production, which enter the intestines, emulsify blood. Some bile salts go back into the intrahepatic circulation, get to the liver, stimulate bile salts. So they keep stimulating their own production. Excess bile gets into the gallbladder. So, yes.
You said that feedback, is that a
Yeah, it's positive feedback. Thank you. Bile is stored in gallbladder. It is concentrated in there. Layers, there's mucosa, muscularis, and adventitia, cirrhosis. Pathway of bile. Will ask you something about it. So know this picture of cystic duct, common bile duct, hepatic duct. Does that make sense? My suggestion, just draw it a couple of times so you can draw it when you get the exam. Just draw it. Just do it at home like a couple of times. It'll stick. Yeah, I kind of answered this already. Yeah. We got mechanisms of gallstones formation. Bile gets concentrated, so cholesterol and calcium form gallstones. Risk factors: Caucasian race, sedentary lifestyle, obesity. Treatment options: remove the gallbladder in its entirety. Pancreas. If I show you on the picture, you identify the pancreas and tail, body and head. Pancreatic juice, chemical component. I refer to bicarbonate. So remember that scheme of bicarbonate synthesis in the pancreas? Know it? Enzymatic components. Table that we had today. Table that you have in your lab. So I'm gonna skip this, and we know the brush border enzymes already, so skipping this too. These questions here, if you filled them out, you may have noticed that they are very repetitive. For instance, this question asks you something that we've already have answers to. Make sense? Like, which salivary enzymes digest carbohydrates? We already mentioned that's amylase. But that repetitive question allows you to synthesize your knowledge, put it all together, and get a better understanding of what's going on and where. Carbohydrate. In terms of, so I have it twice. So monosaccharide absorption. For some reason I have it twice. Now, what do you need to know about absorption? We've done it. Right? That's the part of that study guide review. Does that make sense? Am I clear? Know how these are absorbed. What kind of transport? What is the critical ion, which is sodium? Okay? So, no. This question is actually different. One asks about the apical site here, and one asks about basal site. So, Mechanism of amino acid transport goes to basal side, as Marina mentioned, facilitated diffusion. Okay. So essential driving force, difference in the sodium concentration. Lipids, lipid absorption here to my left, to your right. Know that scheme, that flow chart, okay? Know where they are digested. Why chylomicrons have to have proteins? Because fats are not soluble. So this is now a review. This is why I tell you to work with this study guide, because by the time you're done, by the time you finish filling it out, densely filling it out, you know this stuff. Okay? Now, few words I want to address. Sequence of parts. What's the proper sequence of the colonic parts? Cecum, ascending, transverse, descending, sigmoid, rectum. Do I make sense? Just the anatomical sequence. Know that the only absorptive processes that happen there in the large intestine are ions and water. Commensal bacteria, I mean, we need them. That's kind of bullshit question. Epithelial lining and anal canal. Got it covered today. Structures that support large and small intestine, that's your anatomy question. I'm talking about two amenta, greater and the lesser amenta, and mesentery. Parts of the colon, just mentioned it. Intestinal glands, mucus. They produce mucus. Mold protection. Tinea coli produce mass movements, contractions. Haustra sacs. So they participate in segmentation. Mass movements, large movements of the colonic content that lead to defecation. So slow waves. When intestines is stretched, so stretched will initiate the contraction, because slow waves bring it closer to the threshold of action potential. Peristaltic reflex. Remember that you have stimulatory neurotransmitters released behind the bolus of food. I'm talking about ACH and substance P, and inhibitory neurotransmitters released in front of the bolus. And I'm talking about nitric oxide and vasoactive intestinal peptide, or VIP. And this, so defecation reflex, stretch of the rectum, signal goes to the spinal cord, stimulation or relaxation of the internal sphincter, and conscious relaxation of external sphincter. Does that make sense? Know which sphincter is controlled by which system? Consciously, unconsciously? Voluntarily, involuntarily. Infants do not have conscious control. Vomiting caused by distension or chemical irritation. Sequence of events. Distension or chemical irritation. Signal goes into the medulla oblongata. Medulla oblongata produces chemical stimuli that initiate reverse peristalsis. Sodium, water, potassium. Right there. The mechanism. Now, I very specifically show you which ions I reserve the right to ask about. Calcium. What controls calcium absorption? Which hormone? Parathyroid. Indirectly, right? Indirectly, but parathyroid. Iron, absorbed only when? Yeah. Celiac disease caused by gluten intolerance. So we've got it all